In November 2018, in China, twin girls were born. The identities of these girls, as well as any information regarding their health, is a state-guarded secret. The reason is that they weren't ordinary babies, but the first genetically edited humans ever born. The scientist who performed the gene editing procedure was He Jiankui. When the girls were embryos, Jiankui edited their genes ex vivo, outside of their mother. The goal of the procedure was to disrupt the gene that codes for a receptor on immune cells, which the HIV virus is known to exploit to enter the cells. The edit was intended to make the immune cells more resistant to HIV. The embryos were then implanted into their mother's uterus and successfully born.
And the plan to monitor the children's health for the next 18 years, with the hope that they will consent as adults for continued monitoring and support.
However, nothing except the scientist's claims is known about the girls. The experiment was met by the world with much concern. Editing the genes of an unborn child at such an early stage means altering the very genes in every cell of their organism. These changes will be passed down to their children. This controversial experiment raised a number of ethical dilemmas. Primarily, what if he made a mistake? And what if the procedure will have unexpected consequences? Well, this experiment was in fact illegally performed, and Jiankui served 3 years in prison for such boldness.
The scientific community is that we shouldn't go ahead yet in doing this. So why is there so much secrecy from, for example, the Chinese authorities? So the accusation now is that you've broken the law.
But this video is not about the ethical dilemmas, but about some of the science behind his experiment. You have already probably heard of CRISPR, an insanely advanced gene editing technology, the name of which would make a great biscuit brand. This is exactly what Jiankui used for his experiment. Even though the topic of CRISPR first came up around the 90s, the mechanism itself had been evolving for a few billion years. And originally, it had nothing to do with editing Chinese babies. In this video, I will focus on understanding the original mechanism of CRISPR, and we will see why scientists found this ancient mechanism to be such a revolutionary tool. CRISPR is an extremely effective immunity mechanism in bacteria. The abbreviation is often heard in the context of gene editing, together with the name of a protein called Cas9, aka CRISPR-associated protein number 9. We will go over all steps of the CRISPR process in bacteria. The whole scientific explanation consists of two big chunks. First, a bacterium remembering the virus, and second, using that memory to fight the virus. And then we will connect this curious immunity mechanism to the story of the genetically edited twins. But before I begin the tale, we have to agree on something. The key to understanding CRISPR on a deeper level is dropping the illusion of sentience of molecules. We often hear expressions like looks for and finds in the context of microbiology, but it truly is based on randomness that has been tamed by evolution. Viruses cannot reproduce. In order to reproduce, they inject their DNA into cells of a host organism and have the host organism replicate them. Viruses infect bacteria the same way they infect us, but those that infect bacteria are incompatible with humans or animals or fungi. They're called bacteriophages, or just phages. There are plenty of CRISPR-associated enzymes floating in a bacterium's cytoplasm. Different types of enzymes work with different types of viral genomes or help at different stages of the complex algorithm. I will not be mentioning all of them. We will be discussing the most common case most relevant to the process of human gene editing. Two enzymes called Cas1 and Cas2 capture segments of the viral DNA and insert it into the bacterium's own DNA. The segment captured by the enzymes is not completely random. Other CRISPR-associated enzymes find an NGG motif on the viral DNA, where N is any of the nucleotides. Then, Cas1 and Cas2 cut out a piece of adjacent viral DNA, which is usually around 30 nucleotides long. None of the above happens consciously. Proteins are huge, complex molecules that have unique geometry. This uniqueness is what creates order in the random motion within a cell. Imagine a jigsaw puzzle, where every piece has its own perfect place defined by its geometry. If you were to randomly drag a piece on top of an almost finished puzzle, it would eventually naturally drop into its corresponding space. This NGG detecting step is essential to prevent self-destruction in the future, and we will soon see why. The bacterium that has captured the viral DNA will now pass it on to its daughters as a part of its own DNA, CRISPR region. When the same type of virus infects a daughter, the process of defense begins. Remember the viral DNA the grandma bacterium had captured? Well, that DNA is stored in the part of the bacterium's very own DNA, called the CRISPR region. CRISPR stands for Clustered, Regularly Interspaced Short Palindromic Repeats. It's actually quite simple if you visualize it. The spaces between the repeats are filled with so-called spacers, which are the captured pieces of viral DNA that we already mentioned. They are squeezed between palindromic repeats of the bacterial DNA. The repeats are identical segments of bacterial DNA, and their structure is palindromic. When talking about genetic code, a palindrome means a DNA sequence where both complementary strands are the same when read in the same direction. The direction is determined by the DNA backbone. Let's take a short sequence. GAATTC. The other part of the double helix will be the complementary CTTAAG. One is the exact reflection of the other. This CRISPR region of the genetic code is regularly transcribed. In other words, a complementary negative is taking off the matrix. The negative is called RNA. When this segment is transcribed into an RNA molecule, it looks like this, CUUAAG. And this is where the palindromic structure comes in handy. The piece of RNA can now fold. Because U is complementary to A and C is complementary to G, this type of RNA, derived from a palindromic DNA, folds. Because of its molecular structure, this RNA transcribed of the CRISPR region is cut up into smaller, so-called guide RNAs. These guide RNAs contain the transcription of the viral DNA from the spacer and the handle. This is where the magic happens. There is an enzyme in bacteria called Cas9. This enzyme can't do much on its own because it needs something to guide itself. And this is where the guide RNAs come into play. The Cas9 protein can recognize the geometric shape of the handle and bind to it. Together, Cas9 and guide RNA form a complex, and these complexes patrol the bacterium by simply drifting around. Once the same type of virus, which infected the grandma, infects the granddaughter cell, it eventually bumps into the Cas9 guide RNA complex. These two make a great duo. Guide RNA checks if a part of the viral genome corresponds to its own, because it has been transcribed off a spacer, and the Cas9 protein simultaneously surveys the viral DNA for an NGG motif. When the complex identifies that this is exactly who they're looking for, the Cas9 protein cuts the viral DNA. The cut-up genome is useless, so it no longer poses a threat to the bacterium. Remember what I said about the NGG preventing self-destruction? The Cas1 and Cas2 proteins had specifically taken the DNA segment adjacent to the NGG. The probability of the bacterium's DNA also having an NGG adjacent to the spacer in its CRISPR region is very low. If this NGG control step was missing, then if the Cas9 complex was to bump into the bacterium's own CRISPR region, it would naturally cut it, because the guide RNA would complement a spacer perfectly. This would result in uncontrollably cut bacterial DNA and eventual death. Okay, but why doesn't the viral genome simply regenerate? In theory, it can, but then it would again be targeted by the Cas9 guide RNA complex. Moreover, to repair would need to encounter enzymes that repair the bacterium's own DNA, because two loose DNA ends cannot simply grow back together. And also, right after it's cut, the viral DNA is usually cut up by nucleases, enzymes that break down genome debris in any cell. Therefore, cutting the viral DNA in most cases stops the virus permanently. Let's circle back to the story of the Chinese twins. After understanding what CRISPR initially is, I feel like gene editing still sounds a bit far-fetched. It seems like there's a huge gap between the DNA cutting mechanism in bacteria and the cutting-edge technology that potentially solves all genetically inherited diseases. And while, yes, gene engineering is in fact a whole brave new world, the gap is much smaller than it may seem. For instance, in order to make the twins more HIV resistant, Zhang Kui simply took advantage of the natural DNA repairing mechanism. He cut the target gene by introducing the Cas9 complex, and when the twin cells naturally patched up the cut, the gene was disrupted and could no longer properly produce the protein that the HIV virus so often exploits. But the exact way he did it is a whole different story.